Welcome everyone to the Fermentation Association's webinar of Advances in Yeast. I'm Amelia Nielsen-Stoll, editor of TFA. We are a trade group that was launched to support producers who use fermentation to create delicious and often healthful food and beverages. Our goals are to help educate consumers about fermentation and its benefits, support scientific research into those health benefits, and work with food safety authorities to establish clearer and more appropriate regulations in regards to fermentation. Today, we bring you two great speakers, Richard Preiss, co-founder of Escarpment Laboratories, and Dr. Ben Wolfe, associate professor at Tufts University. We have many questions already submitted and reviewed with our speakers. If there are additional topics you'd like to see addressed, please enter them in the chat below and we will try to get to them. I will come back at the end to ask some of our audience questions. For now, I will turn it over to you, Ben.
Great, thank you so much. And thanks everyone for coming out today. Really excited to have this conversation. So as Amelia talked about, I'm on the advisory board for the Fermentation Association. And what we're doing is really thinking about ways to open up discussions with various stakeholders, academic, industry, folks who are at home or just enthusiasts of fermentations and fermented foods, to begin to understand what are some of the most cutting edge and exciting advances that are happening in the world of fermented foods. And so today, Richard and I are going to be talking about one of those areas, and that is the area of yeast. And of course, yeasts are important in a lot of different fermented foods. And I'm really excited to be able to chat with Richard because I think of him and his group as sort of a pioneer in really thinking about where we are and the technologies that we have in terms of yeast, but also where we will be in the future. How is this all moving forward in the next 10 years or so? So a little bit more about Richard and why I'm really excited to chat with him. So Richard is one of the co-founders of Escarpment Laboratories in Guelph, Ontario. And I got to know Richard through sort of the online Twitter community as someone who was working on his master's at the University of Guelph, where I also have a master's in botany, on molecular biology and genetics. I got really excited about yeasts there. And then started this really exciting company, Escarpment Lab. And for those who aren't familiar with the terrain of Ontario, escarpment refers to this sort of series of cliffs that spread across Ontario. So that's where the name comes from, and the really cool logo as well. And so what I love about Escarpment is, they're providing some basic services in terms of strains of yeast largely for beer brewing, but also for other types of fermented beverages and foods. But also they're doing a lot of exciting R&D and thinking about how to innovate in the space of yeasts and yeast for fermented foods. So what Richard and I will be doing today is really more of a conversation, less of a formal lecture, because lectures are kind of boring sometimes, and we thought this might be better covered in just a chat back and forth. So thanks everyone for coming out. Richard, do you want to introduce yourself a little bit more, talk a little bit about Escarpment and how Escarpment got started?
Yeah, first of all, I want to say thanks for that. That's an awesome intro. And I don't know if you remember this, Ben, but our history actually goes a little bit further. So I emailed you at some point in my undergrad because I was really interested in the work that you and Rachel Dutton were doing with cheese and microbes at Harvard. And I was really curious about some of those questions around terroir and things like that. And I emailed you out of the blue saying, like, how would someone actually prove this? And you responded in detail saying, this is how someone would do this, but it would be a full, PhD project. You took the time to respond to me when I was, in an undergrad curious about microbiology. And, I want to thank you for that, because that was, one of those things that inspired me to keep on this path. So I think you can take a little bit of credit for
Awesome.
As far as Escarpment Labs has gone.
I'm glad I replied back to that email.
Yeah, I don't still have that, but it was really, really nice to, it's always those kinds of things, hear back from people that are experts in these spaces, with their advice or their ideas. That's the thing that I love about science, is that it's so collaborative, and including the business that we're in as well, working with brewers and other fermentation enthusiasts. It's this incredibly open and collaborative space compared to a lot of industries, and I think that that's like our secret weapon or our secret value is that fermentation is so open in terms of access to knowledge, as well as in terms of just, people being willing to experiment and try new things. And that's how it's able to develop so quickly. So that's really the focus that we're taking at Escarpment Labs, is keeping an open mind and sharing whatever we learn along the way, because that's going to help all the people that we're ultimately supplying cultures to. It's going to help them make their products better, be more profitable businesses, or just have more fun experimenting at home.
I really love that point, because I think in terms of thinking about advances across fermented foods, not just with yeast, but with other microbes as well, I do think that this sort of open collaborative idea is very strong across all fermentations, especially in North America, where I think fermented foods are so new and we're still figuring them out. And there's so many new producers that, people are doing this at the scale where they need to work together with other people. We need to be collaborative. So I really like that point. I think that's a really great idea. Yeah. So I guess let's start off with this conversation and thinking about, really big picture from your perspective. And then I can chime in maybe from the cheese and sourdough and other worlds that I work in our lab at Tufts. But what do you think have been some of the really big transformational sort of technologies that are changing how we think about yeast in fermented foods?
There's been a lot of change, especially in the last 10 years, 10, 15 years. It's kind of amazing how fast things have changed and are changing. And it's really, I think it's really fun to be a part of that change. Honestly, number one to me is the rate at which genetic sequencing, genomic sequencing has decreased in cost. And I don't think this is something that is common knowledge, but you can get a good quality genomic sequence of the whole yeast genome of whatever it is you've got in hand, and you want to find out more about it. You can get that whole genome for $200. The tools to deal with that data and get some meaning from it have never been more accessible as well. And I think that's incredibly powerful, because this is something that used to cost thousands, or in the case of the first yeast genome, millions of dollars, and the rate at which the cost has decreased, I think, is a substantial change that we've encountered. Even in my time in doing yeast science from 8 years ago to now, that cost has gone down, and we're at the point now where sequencing a yeast and basically learning everything we can from its genome is, maybe 5 or 6 days of processing and $200 away, and that is incredibly powerful.
Yeah, I agree. I think that the way that we think about genomic data now and what we can do is incredible. So when I started my PhD, it was just when DNA sequencing was becoming what we call high throughput, where you could do it really quickly and relatively cheaply. And now the things we do in my lab, even back when I started my PhD, would have taken weeks or months, where we do it in a couple of days. And I tell everyone in my lab, you are so lucky to be able to get this much information so quickly. And I can, pay for it, without a problem, because it's so cheap. So I think most of our conversation today, I bet will sort of focus on that genomics aspect of yeast and fermented foods, just because that's really been where a lot of the transformation's been at. Yeah,
for sure. And the other thing I would sort of add to address that question would be, there's also been a lot of work on discovering new genetics, right? Going out into other fermentation environments and other places and asking the question of, what yeast is there? What do they do? What's special about them? And we've seen some cases where that has created a lot of value. I can think of some of the work, and I think you've been involved in some of this, with sequencing sourdoughs and figuring out what yeasts are living in sourdoughs and what's special about them, what makes them different from industrial baking yeasts. And that's very interesting, because they do have some clearly defined differences. And there's some of the work that we've been involved with in identifying some yeasts from traditional farmhouse brewing that's going on in places like Norway. So you hear this a lot in the beer side, people talking about this Kveik yeast. It's a traditional yeast that people have kept alive in Norway, and we were able to take it into the lab and do some studies on it and found that it had some really interesting traits that other beer yeasts weren't offering, and it was just kind of sitting there. So I think that's another. I mean, maybe this isn't a technological advancement, but I think there's a lot of curiosity in yeast that has caused people to look around and see what else might be out there that still needs to be defined or discovered.
Yeah, and I think, you know, in one way, the genomic sequencing is great for basic biology, right? It tells us what the genetic blueprint is for all these different organisms. And that allows basic scientists like myself to say, how are these things related to one another? But at the same time, having that sense of organization, I think, is a really powerful tool for people who are doing fermentation, right? Because if we just have all these strains out there, we don't know how they're related to each other, or how a new strain you might be getting from the wild is related to existing strains. It takes a long time to really figure out that strain. Whereas now, when you can sequence it and get information back in like a week to know how it relates to, you know, traditional strains that have been used for decades, that might be really powerful for thinking about the next steps you might go with that strain. Yeah, yeah.
Yeah, there's a lot of potential for, you know, using some of that information to cut out all the trial and error, or a lot of the trial and error that otherwise has to be done.
Exactly, exactly. So one thing I think would be really cool to hear about is maybe a specific technology or application that you're using at Escarpment, maybe along those lines, where you're using genomes or genomics or genomic sequencing to really help you figure out some new strain that you might want to develop.
Yeah, we try to jump on any opportunity to use new technology, especially when it comes to, you know, opening up some collaborations with some of the, you know, university researchers that are really good at this stuff. So, you know, in one case, we've got a project to use genome sequencing, high quality genome sequencing, combined with just some plain old screens of a bunch of beer yeasts, and then basically trying to take that data and put it together and get some meaning from it, right? You know, there's still a lot of open questions with our brewing yeast, or just with yeast in general. Like there's certain flavors where the underlying genetics aren't very well understood. So what we're hoping there is that, you know, if we take the flavor profile of all of these different beers made with 40 different yeast strains, and we take their genetics, we can start to look for connections between those things and understand those better, and understand, you know, maybe this combination of 12 mutations is the underlying, you know, influence for a certain trait that might otherwise not have been so obvious. So, you know, being able to sort of tie the genotype, you know, what the code says, to the phenotype, which is what the yeast does better, is a focus for us. And then, you know, even beyond that, having a better understanding of that link then allows us to, yeah, if we get an unknown yeast strain, we can get a sequence, and we can, you know, hopefully in the future have a better prediction of how this thing's going to behave, so we don't have to do as much trial and error in the lab, because, you know, it's fun, but fermentations take a lot of time. And then moving beyond that, understanding what each yeast strain, you know, kind of has in its code, we can start to look at things like yeast breeding, which that's not new technology, but there's still a lot of potential that hasn't been realized. You know, taking one strain and literally breeding it to another strain to yield something that's somewhere in between. There's definitely a lot of potential there. And then, you know, maybe downstream, this is not something we've got into yet, but, you know, utilizing some of those unique genetic features to deliberately reproduce them, like using a technology like CRISPR is possible as well.
Very cool. Yeah. So maybe I'll step back and just show some folks some pictures, some slides. I said I wouldn't lecture, but I think sometimes illustrations are really helpful. And just in case folks aren't on the same page in terms of what we mean by genome sequencing, this might be really helpful. So I'm going to just share my screen really quickly, and we can sort of nerd out about some cool data that other people have already made. So this is just a quick overview of what genome sequencing can look like. And this is not going into the nitty-gritty details. This is sort of a high-level view. And again, this is just to get everyone that's out there listening on the same page. So this picture I have here on the left-hand side is just a plate of, this is actually from a cheese rind, but this could be from any fermented food where you grow up the microbe on a petri dish. And these little white colonies here, those are yeast, which of course is what we're talking about today. And then in order to get that genome that Richard and I have been talking about, you have to get the DNA. So the blueprint, the genetic blueprint that you need for all materials for life. So in this case, I have these little cartoon sticks to represent what are called the chromosomes, which contain all the DNA. And remember that DNA is made up of these different letters when we think about the genetic blueprint, these different bases, the A, T, C, and G. Then what we do, and there's a gazillion of different ways to do this now, we sequence that DNA. And we can talk maybe later, Richard, about the various technologies that you're using at Escarpment, but there's a lot of different companies that offer DNA sequencing. And what I'll point out is that this same sequencing, the same approach is being used right now for tracking the coronavirus, right? So all these variants that you hear about in the news, those variants are variants of sequences. And it's not necessarily DNA always when we're talking about viruses, but it's the same overall approach where you're trying to figure out the genetic blueprint of an organism. We're talking about yeast, but the same thing can be applied to the coronavirus. And then what you get back at the end of the day is this overall draft genome. So this blueprint that shows you all the different genes that are contained in that organism, how they're related to each other in terms of their locations across the DNA. And sometimes when you do what's called annotation, you can get guesses as to what those genes can do. So the actual potential function. So maybe it's a gene in a yeast involved with making alcohol, or maybe it's a gene involved with making various kinds of flavors. So this is like a very crude overview of what genome sequencing can look like for yeast, but for any organism as well. So one example, going back to what Richard was talking about in terms of the utility of genome sequencing, like why do this, is just to sort of organize the diversity of yeasts. And this is an amazing example. I think we've read this paper multiple times in my lab. And I know Richard is a big fan of this paper. Many people are a big fan of this paper. This is coming out of Kevin Verstreppen's group, who is over in Belgium, and many other collaborators as well. And what they did in this paper is they went out and just sequenced a whole bunch of different yeasts, so the entire genome of those yeasts, and said, how do these different yeasts relate to each other, right? So how are they connected to each other based on their genomes, based on how similar the sequences are of their genomes? And they were particularly interested in beer yeast in this paper, but they included some other sort of non-beer yeast to be able to organize that. And I think they made this like super awesome picture that I want to hang up and have as like a poster in our lab because it's so beautiful. And this is actually an output you would get for a lot of different types of genome sequencing that we're talking about today. And what this is we're looking at what's called a phylogenetic tree. And so it's a tree, so just like a tree out in the woods, this tree has tips and branches. And in some cases, the tree can actually even have roots. And so the way to think about this tree, it's in a circular form, but each of these little branches here, each of these tips represents a different yeast strain, so a different isolate. And this paper was specifically thinking about Saccharomyces, which is the baker's yeast or brewer's yeast, but you could do this with any kind of yeast. And the way to think about a phylogenetic tree, it's a lot like a family tree and how you're related to your parents and your grandparents and your siblings. The closer these strains are to each other in the tree, so the closer the branches are to one another, the more similar they are genomically to each other. And so what we're seeing here is how a whole bunch of different beer yeasts, so you can see there's a group of beer yeasts over here. There's a huge group of beer yeasts over here that includes things from Belgium, the US, and Britain. And then we can also see other types of yeasts. So you'll notice there are yeasts here from Asia. There are yeasts here from wine. There's sort of mixed fermentation over here. And these are coming from different places. So for example, a lot of these Asian yeasts are from sake. So this is a Saccharomyces species that's used in the production of sake in Japan. And so what this does, this big tree of yeast, it helps us take all this biodiversity that's kind of out there, but we don't necessarily know how it's all related, and organizes it for us and says, for example, these are one group of beer yeast that are really genetically related to each other, and they're actually quite different from this huge other group of beer yeast that we have over here. And so this is just one example of how fermented food yeast sequencing, in terms of genome sequencing, can organize what is otherwise a big mess into a sort of clear picture for us. And you can read more about this paper, the Cell paper, the journal Cell that it came out in. I think it's actually a really clearly written paper. They do a lot of great work trying to connect this genomic information in this family tree to what the yeast can actually do in terms of flavor production, tolerance of alcohol concentrations, tolerance of acidity, and other things that are really relevant for fermentation. Richard, do you have any other thoughts about this particular figure and this sort of overall effort to organize Saccharomyces diversity?
I mean, the only thing that I would add, and I don't want to make things too confusing, is that because Saccharomyces is really good at breeding with other Saccharomyces, some of these branches are not necessarily, linear. You have some history of what's called introgression, or, mixing between groups. And there has been some research since this that has shown, some evidence for, different points of introgression. So from what we can see of the genomes, it's a bit more of a twisted tree, tangled tree than this shows. But I think this is a really good way of, kind of simplifying what's going on. But it does get really complicated, especially for, those of us who are doing some of this yeast genomics work, because sometimes you encounter something that just looks very strange and it needs to be explained by, some event that happened 1,000 years ago, and all we have is genetic data. So it gets pretty tricky.
Yeah, and we can talk maybe more later about the limitations of genomes, right? It's only one piece of information, which I think is a really, really great point. Mm-hmm. But so I think this is a great example of how we can use genome sequencing just to sort of make sense of what's out there. Another example of this actually includes, this is from a paper that hasn't been published yet, but this is from Maitreya Dunham's group, and she's been working on a really cool project looking at the evolution of yeasts in breweries and thinking about how that might change over time. And again, you can use genome sequencing to do that. You can actually look at mutations popping up, just like mutations are popping up in the coronavirus as it spreads across the world. Mutations can pop up in yeast as they're being used in breweries, and lots of different types of mutations. But one of the things they did first was just sort of organize diversity of strains that were being used here. And what I love about this is we're actually seeing one of the escarpment strains. So Richard, do you want to talk a little bit more about what these particular strains are that they were sequencing, or what this strain is of your escarpment Cali ale?
Sure. It's a really cool project because, like you said, they were asking, what evolution is occurring in a brewery. So they were able to do some experiments collaborating with a brewery. I think it was called Postdoc Brewing Co. Yep. And basically measure and test what changes happened in the genome over dozens of generations of reusing the yeast. So really, really cool study. And, as part of it, they also got as many yeast samples from this yeast family as they could. So this is what a lot of brewers will call Cali or Chico. Very, very common clean ale strain used for beer fermentations. It's one of, I would say, maybe one of, if not the most common yeast used for craft beer, especially ales. It's an ale strain. So it's really cool to see this kind of deep dive on the genetics of this family as well, since it's such an industrially important group of yeasts. And it's, interesting seeing how all of the different commercial options, from different yeast providers or isolated from different breweries, relate to each other. And, in some cases, there are some interesting mutations that have that differentiate them, even though, they're on paper the same yeast.
Yeah, and again, this may not be something you maybe would know that the strains would behave differently from one another, but you wouldn't know why. And so this approach can help you sort of begin to understand that. And so what we're seeing, this is another phylogenetic tree, so another family tree of these yeasts. And again, each of these little boxes with the naming here is a different strain of yeast. And the one thing to note as you're looking across this, you see some colors that are changing. So the blue and the green here. And this is actually referring to the number of variants or copy numbers of a particular genetic region in this particular strain. So some of them have 3 copies, some of them have 4 copies. And again, these genetic differences across strains can sometimes lead to huge differences in important fermentation properties. So things like tolerance of particular stresses that they might encounter during fermentation, or even sometimes flavor production as well. So yeah, so you can check out this paper as well. This is it's still in review, but it's out there in the world on something called bioRxiv. And it's a really cool paper that demonstrates the power of genomics. So yeah, I'm gonna stop sharing and come back and continue this conversation a little bit more. Richard, how is genomics being used out in breweries, other than that really cool example that we just talked about with Maitreya? Is it more of like something that very small labs are doing in isolation and it's not really being applied yet? Or is it still just basic science, or are cool applications happening that you could talk about?
I think that's where we come in. The yeast providers or yeast scientists can come in. And that's also where there's a ton of opportunity, I think, in the future, because craft beer is so collaborative, and at its best, so is academia, and like there's a lot of opportunity, I think, for these kinds of collaborations in the future. And we've seen a lot of these kinds of things happen, and a lot of benefit come from it. So it does take someone to translate that knowledge or convert that knowledge, from basic research into some kind of application. So that is where, companies like Escarpment or other yeast companies can come in and, work with those end users, those brewers, right, to make use of that information. Because if it's sort of just sitting in a silo over here, it's not doing anything. It needs to be basically let loose. It needs to be also translated, because I don't expect a brewer to know how to sequence a yeast genome or read a phylogenetic tree. They just want to know, how am I going to make better beer? Right. How am I going to, be less stressed out about my fermentations, right? And so there's this kind of space in the middle where we need to come in and make that information useful, right? And where we've seen that be helpful is having that basic science information informs how we can provide advice or select strains or troubleshoot problems with the brewers, right? Having some of that information makes this easier for us. Just sort of thinking of some examples. A brewer might be having a problem with some kind of stability of some kind of yeast phenotype, right? Like the common one is the flocculation changes of the yeast. So something that previously was really flocculent, settling out to the bottom of the beer, leaving a clear beer, is suddenly not, or vice versa. And like there's different cases where a brewer might want the yeast to settle all the way out, or they may want a little bit more in suspension for some of the like hazy IPAs and things like that. So we know things about the flocculation genes of those yeasts, and we can use that information to inform how we might help a brewer troubleshoot and ultimately make their fermentations more consistent.
Yeah, and I think that's a really great point too. I've worked with cheesemakers or other fermented food producers who have yeasts that are maybe wild yeast that are just floating around their facility. They're behaving in a particular way for a long period of time, then all of a sudden they stop behaving that particular way and they start doing something else. And so one question could be, is there a new yeast present? Maybe there was some invasion of a yeast that came into that fermented food facility, and now that yeast has taken over. Or, an alternative explanation is maybe the yeast that was already there has evolved. Maybe some mutation has popped up, and all of a sudden it's starting to behave differently. And so that's another great example where for any fermented food, not just for beer, but for any fermentation that has yeast, where you have the same microbes around for a long period of time. So sourdough, kombucha, and in some cheese facilities, that's where genome sequencing come in and kind of at a very fine scale, look for mutations in the DNA that might explain these changes. And I guess the one question is then what do you do, right? So once you know that a mutation caused the problem, how do you fix the problem, right? And that's honestly where a lot of basic scientists like myself don't necessarily go all the way, because we can't always figure out how to make that change. So yeah, how do you make those changes once you have the genomic information, or how do you change your management strategy?
I mean, if something has changed in terms of the microbes in a way that it's not working the way that it used to, that is I think an easier problem for a yeast company, a culture company to solve, right? Because that's our bread and butter, is keeping a stable collection of microbes that the brewers or the fermenters have access to, right? So that would always be my approach, is say, for example, we're monitoring yeast in a cheese facility. Let's get as much of the stable or desired microbiota as possible, and make sure that we have it preserved, because then if things go haywire, what we can do is we can grow some of those things up and try to bring it back into balance. And it's a similar approach in the brewery. I would say maybe a little bit simpler in that case, because the brewer can just stop using that yeast and order a new batch. So that's an easy problem. But there's some scenarios where we have brewers that are using mixed cultures, for example, where that's not desirable. So then having that original culture or stable culture as like an insurance policy then becomes extremely valuable, because then you can go back and, for example, if it's a sour beer culture and it's just getting way too sour, we can go back and grow up the yeast and try to balance it out for them.
Right. Yeah, so I guess another thing that I hear a lot of people talking about using genomics to do is to go out and find novel yeasts, right? And sort of do hunting expeditions for yeast, and sequence the genomes, compare it to strains we already have, and see if they'll be good or not. That seems to me as an overwhelming task, right? Because how do you first find those yeasts? How do you know which ones to sequence? And even then, once you have the genome, how do you know which of those that you sequence will be useful in some kind of fermentation sense? Does Escarpment have some experience of sort of going out and hunting for wild yeast and using genome sequencing to make sense of anything that you find?
We do have lots of experience hunting for wild yeast. That's always been a big part of what we do. Originally the company was started on that idea of offering local wild yeast. We quickly discovered that most brewers just wanted basic Cali ale. And so we kind of had to pivot to offer that, or offer the basic yeasts, but we still do it because we still see a lot of value come out of looking in the wild for yeast. I will say that we're not really at the point of using genomic tools or genetic tools to make that process easier, although there is potential for that. And there are some ideas in the works there, but how it works for us is still pretty analog. Take something from the wild, tree bark and fruit are great places to look for wild yeast that'll end up fermenting beer or other fermented things. Soak it in some kind of nutrient-rich growth medium, and then basically plate it out onto those agar plates like you showed in your slides. Get something in pure culture, and then screen it. And screening is still a very useful tool. Basically, just take everything you find and throw it at some kind of hurdle and see what kind of gets through. And that's kind of been our approach, is basically putting anything we find in the wild through a series of hurdles, and then whatever we end up with it at the end is probably somewhat useful for whatever target we've set, whether it's fermenting beer or wine or whatever the goal is. And then from there, that's usually where the genetics will come in. And okay, what is it? Is it safe for consumption? Based on the species, do we know anything about it? In some cases, with some of the wild yeast that we find, there's very, very little information out there that we can make use of, which is a challenge. In other cases, they're very, very well characterized yeasts, or kind of like up and coming yeasts where there is some information that says, oh, this yeast is actually maybe useful in fermentations because it can make lactic acid or something like that.
Right.
So that's kind of been the approach. I think in the future, it would be my dream to be able to just, take that liquid that a piece of tree bark has soaked in and do some sequencing and figure out if anything useful is in there before we do all the extra work. But we're not there yet.
I mean, that actually brings up a really great point, that with the exception, I'd say, for maybe beer and wine, our just general databases of genomes for yeast is pretty poor for many fermented foods. So I'm thinking especially things like kombucha, some funky sort of sourdough yeasts, and a lot of other fermented foods. So there are actually yeast in kimchi and sauerkrauts and a lot of other vegetable ferments with names like Kazakhstania and Wickerhammer-Myces, which are, they kind of get a lot of the less of attention because Saccharomyces gets all the attention in the yeast world, but they're out there and they're really important. But we actually are lacking a lot of genomic data for them. So I think, moving forward, labs like mine and other labs that are interested in these, the more genomes we can collect from those and begin to characterize their DNA sequences, but also their phenotypes, like we talked about. I think the more the whole field will advance in that sort of collaborative sense we were talking about. There really are a lot of yeasts that have been neglected historically.
Yeah, absolutely. And again, like information in one place is useful in other places. Like a lot of the like, especially with the wild yeast that we find, there's usually not much research on these things in beer. There's maybe something about using Hanseníaspora uverum or something like that you find like in wine or cider or something like that. And we kind of have to try to translate that information. So yeah, I do think that there's a need to just keep looking at what yeasts are popping up in different types of ferments. Like we've found some yeast in lacto ferments that were pretty interesting, right? So there's a lot of potential out there.
Yeah. So I think a couple of caveats to all of this. We're talking about the wonders of genome sequencing, but and I've seen some questions popping up in the chat about this. There are some limitations, right? There are some big limitations. One is that a genome just tells you what an organism can do, right? It tells you its genetic potential. It doesn't tell you that it will actually do that, or when it will do that, or how it will do that, right? It's just showing us the sort of the potential for doing various things, but not exactly how it does that. And then I think there's some other limitations in that, we're thinking about just single organisms, but of course, most fermented foods, it's not just one genome, it's a collection of many genomes that are interacting in so many different ways. And that can change how each of these organisms functions. Any other limitations you can think of in terms of using genomic data?
Well, that was the one that I was going to, highlight, is that in some of these cases where you have multiple microbes, we first of all, we don't know how the individuals are, who they are, or how to speak their language. Second of all, we don't know how the heck these things communicate with their neighbors, right? Like, and that is a huge gap in our knowledge with microbiology in general. And I'm thankful that people like you exist with research programs focused on microbial communities, because like this is a huge knowledge gap, because we've been focused on isolating microbes and their behaviors for 150 years. And we've probably missed some things along the way in that process.
Absolutely. Yeah. And every time we sequence a new genome or put organisms together, it kind of changes everything we thought we knew about those organisms. So it's a very exciting place to be working right now. So one thing that I've definitely encountered applying, so fermented foods are these very traditional things that we've been making for hundreds of years, sometimes by accident, right? And they come in with this DNA sequencing, and we think we're going to like change everything we know about it and maybe how we ferment. So there's sort of this like tradition versus technology conflicts that can emerge. Do you think that there are some places where we don't need DNA sequencing, or we don't need to innovate, or maybe even that having all this genomic knowledge might take away some of the mystery and the magic of fermented foods?
I don't think it's necessary, no. And I do think that like understanding and respecting tradition is a key part of this, because in a lot of cases, first of all, like the tools, the microbes, but also the techniques that people are using in traditional fermentations is often a reason they exist. And these practices have been tested for hundreds or thousands of years, and they cannot be dismissed, right? So I think that that's, first of all, really important, is that there's a lot that the science can learn from the traditions. And, I'll talk about those Norwegian yeasts again, the Kveik yeast that these home brewers have basically been keeping alive out in the fjords. These outperform every industrial brewing strain that we've compared them to. These are scientifically superior to anything that thus far that anyone that I've seen has come up with in a lab. So, in some ways, the traditional products can even outperform anything that we come up with. So, no, it's really important to pay attention to tradition. And that being said, I do think that there's still a place for innovation, despite such a long history of tradition with fermentations, right? Like the history of humanity is kind of intertwined with the history of or the story of making fermentation better. And that includes science, right? Like a lot of the key advances in science are literally a result of people trying to make fermentation better. Like most of the stuff that Louis Pasteur was doing was him trying to make wine and beer makers not do dumb things. Carlsberg, in the process of making beer better, invented the pH scale. They discovered enzymes. They did, a whole lot of pure cultures. They did a whole lot of, really important things in basic science along the way of trying to hone in this traditional fermentation.
Yeah, I tell my students when I teach microbiology that much of the history of microbiology is food microbiology, is actually food microbes, and they really drove the innovation of the field. So it really all comes back to food and fermentation often. Yeah. So in the same vein about sort of technology and tradition, one thing that comes up a lot when we're talking about genetic sequencing are genetically modified organisms. So thinking about using genetically modified microbes in fermentation. So yeah, I think it's a really interesting issue. I think in many ways, GMOs and genetically modified microbes might allow us to innovate, right? And get completely new flavors in some traditional fermented foods. But of course, there are many people that have concerns about GMOs in foods in general. Do you have any sort of thoughts about GMOs and in brewing particularly, maybe?
Yeah, I mean, that topic alone, I think we could talk for hours.
Whole other session.
Yes, I'll try to cover some of the basics. Yeah, like there is I think there is potential for GMOs, for introducing, deliberately introducing new genetic features. That's how I'm going to define it. Deliberately introducing new genetic features into a yeast to make a beer. And we already see some of that being explored, especially in the States, where, there are a few companies offering genetically modified yeasts for beer that have functions that a regular Saccharomyces cerevisiae yeast might not otherwise be able to do. So for example, a company called Lallemand has a product called Sourvisiae that makes lactic acid. So they've engineered a pathway for producing lactic acid into that yeast, so it produces lactic acid and ethanol. So the brewer doesn't have to add bacteria to make a sour beer, for example. And then there's other examples. There's a whole company called Berkeley Brewing that's basically built their value proposition around genetically modified yeasts, and they're making some things that are interesting as well, like plugging in flavor production pathways from plants, so that you have a yeast that produces terpenes, some of those really volatile, expressive aromas from plants. So you can have a yeast that smells like lavender or smells like hops and things like that. And that's kind of interesting as well. So I think that there is a place for it. I do acknowledge, especially for our position in craft beer, like novelty does have value. I think some of these things are novelties. Some of them might stand to become long-term tools that are used in the industry. But yeah, it's been interesting to me to see how readily the craft beer industry has sort of adopted the ideas of GMOs. Like, I think I had been saying this for a few years, like craft beer is going to be the first food space where the use of GMOs is widespread, the first craft food space where the use of GMOs is widespread. And we're seeing that play out a lot faster than I thought it would, with some of these products already on the market available to brewers in the US, and they're using them.
And I think maybe the one big difference between something like wine or beer versus something like cheese or sauerkraut is that at the end of the day, the microbes are dead in a beer, typically. So you're not actually consuming a living GMO versus in cheese or something that's considered probiotic where the microbes are still alive. You actually are consuming those genetically modified organisms. So maybe that's where there's a divide that develops in thinking about where these things might be more readily used versus other places.
Yeah, that's a fair point. Like the yeast is usually removed. I mean, the other thing that I would add there is that with microbes, the whole GMO question gets tricky because they've done this stuff naturally. You know, microbes pick up genetic material, useful genetic material from each other all the time, and yeast is no exception to that. And a lot of the times, if a yeast is picking up a new piece of genetic material from some other yeast species and it gives it a new function, it's the exact same thing that a human making a GMO is doing can also happen in nature. So that's something that we should recognize as well, is like this stuff can happen naturally. Like the yeast that is commonly used for champagne production has picked up a bunch of genes from a different yeast species that make it better at thriving in that environment. And it's not a GMO, but the same transfer of genetic material happened naturally, you know, in a fairly recent timeline.
I think it's a really good point, because I think a lot of the papers that are coming out right now on yeast genome sequencing, a huge theme is how much genetic material they're picking up from other species. And for folks who aren't familiar with this space, it's something often called gene transfer. So this is where bits of genetic information, sometimes it's a whole gene or a part of a gene, are taken up by an organism and get integrated into their genome. And then they actually can start using that gene and the proteins that that gene can make for their own biology. And a lot of this is just by chance, right? A lot of this is just sort of part of the evolutionary process that all microbes go through. But, you know, work from my own lab and from other labs in cheese and sauerkraut and all these other fermented foods are showing so much genetic exchange that's already happening. So maybe we can think about those quote unquote natural processes that are happening, and maybe instead of, you know, using more advanced GMO techniques, we can just sort of tap into some of these more traditional microbially mediated processes to get some of the innovation that we're looking for. It's a really interesting area to see in the future. So speaking of the future, if you could get out your crystal ball before we break out into some questions, what do you think the future of yeast fermentation looks like from the genomics perspective, or maybe other technologies that might come in and disrupt this space? Like what does the next 10 years look like for you?
Yeah, I have a list of things written out here. We already touched on novelty. I do think that, you know, using breeding or genetic modification to produce yeast that can do things that they haven't done before is going to be a trend of the next 10 years. You know, some of those will stick around, some of them won't. For sure, you know, craft beer, at least, you know, our industry does thrive on novelty. So, you know, I think that's a reality. And I do think that, like, in general, we're going to see an acceleration in both the acceptance and the use of biotechnology. I think, you know, I've heard people talk about how COVID accelerated e-commerce by 10 years because everyone was shopping online. What I haven't heard people say as much is how much COVID is going to accelerate our acceptance of biotechnology. The average person knows what PCR is. The average person knows what using recombinant DNA technology to make a vaccine does for them. They know the benefits of it. They have lived that. And I think that also, you know, living through COVID and recognizing that the solution to COVID has been this kind of science and biotechnology is potentially going to accelerate the acceptance of some of those biotechnologies, you know, things like genetic modification, for example, faster than we might have otherwise considered. So I think that's going to really frame things for the next 10 years. I also do think that we're going to start seeing, you know, other external pressures become important again. You know, something we haven't talked about for a couple of years is climate change, and that's going to become relevant. That's going to come roaring back into focus because it hasn't gone away. And we're going to start hearing about, you know, some of the approaches that need to be taken in our food systems to address and protect ourselves against climate change. So that might be things. There's really cool stuff on you know, the plant side, on breeding plants that are more drought resistant, for example, and, you know, barley and hops are, you know, grapes are no exception. But, you know, we might also see further use of yeast technology to reduce the need for cooling, or to increase yields, or increase the rate of fermentation so that brewing facilities can become more efficient with their energy. So I think that's going to be a bigger story.
That's a really great point too, because so many fermented foods, there are wastes, right, that can come out of fermentation. I think of dairy, you know, yogurt and cheese production. There's a lot of whey and other wastes that come out, and yeasts are often very simple organisms that do really cool things to help deal with those wastes. So I love that idea of yeast not necessarily just being at the forefront of innovating for flavor and for quality, but also for sustainability in fermented foods. That's a really awesome point.
Yep.
Yeah.
Craft beer is massively wasteful in terms of the things that get thrown away and the amount of water that gets in energy that gets used. So it is, I think, something we're going to have to reckon with the next 10 years.
Cool.
And then the other thing that I think is fascinating is, again, you know, the genomics getting cheaper, the ability to analyze that genomic data and make use of it getting more and more accessible, and then also seeing some of the access to actually designing new things or even you know, massively breeding yeast. Like you see companies like Ginkgo that are basically setting up a platform for companies, smaller companies that don't have access to millions of dollars of robots, suddenly making some of these projects accessible. And I think that's going to be a huge development as well, because, you know, you might have a company that is able to develop solutions for problems without having to invest millions of dollars in labs and robots and all these things, right? If a company just says, hey, I want to remediate some contaminated soil, you know, here's the soil and here's some microbes that I think we can develop to address this problem. I think a lot of those kinds of things, developing and adapting microbes to solve problems, and yeast is no exception. I think a lot of that's going to become a lot easier as we have these like larger scale tools to do things like synthetic biology or genetic modification.
This is great. It's an exciting future. So hopefully there are more people like you that'll send me emails and I can have a larger group of people to recruit and have as part of this exciting yeast revolution. This is awesome. So I think what we'll do now is break out with Amelia to get some questions that have been popping up in the chat and sort of some of the themes that she's seeing there.
Thank you so much, you guys. This has been so interesting. Really, the sequencing is opening a whole new world of fermentation, which is really exciting. So let me get to a few of these audience questions. We had one, so as yeasts are becoming increasingly genomically sequenced, how is this impacting intellectual property? Are people patenting yeasts, especially when a specific strain of yeast are quite valuable in food or beverage industry? How does that work when a wild yeast enters a lab?
That is a really awesome question and a really murky area. I might hand it over to Richard from his own experience thinking about how to sort of protect the property and the money spent on R&D for these kinds of creatures. Yeah.
People are trying to patent natural yeasts, and sometimes they're successful. I'm really not a fan of that practice. I really think that it's not very constructive. I also think it's pretty problematic if someone can find a yeast from the wild and not make any changes to it and say, I own this now. Not cool. There is at least one case, for example, of a yeast species that you can easily find in the wild that has been patented for making beer, for the application of making beer. That sucks because now it means that no one else can do this without infringing on this company's patent. There are some other cases where these things have been, people have tried to patent. I talked about the yeast that makes lactic acid. There's yeast that do this naturally, called Lachantia, and a couple folks have tried to patent using this thing in beer, and thus far those have been reduced in scope, which is a good sign. But yeah, like it's tricky, it's murky territory, because I do see the need, especially for universities, to protect the time and money that's been invested in the research. But on the other hand, like in some of these cases, there, in my mind, is not a lot of value actually being added by the people, by the researchers, in terms of this actual invention. So it gets to this kind of funny area. If someone's finding an interesting feature from a wild yeast and plugging it into Saccharomyces and patenting a method on doing that, yeah, absolutely. It's very clear that you've done something. But if you're just finding something from nature and demonstrating what it does, then I struggle to define that as an invention.
And I agree with that. I feel like in many ways, you have to spend the time to somehow change or modify or develop a process to use the yeast. You can't just go out in nature, find it and say, this is new and different, I want to claim this. But again, it's tricky. It also depends on country that you're in. It depends on the particular food and application. And I'm not a lawyer, so I don't necessarily know all the answers to this. But I think going back to Richard's really early point in this conversation, that keeping yeast and fermentation and microbes and fermentation as a collaborative, sort of open community effort in the long run will be best for everyone. And I know there's always, the balance of profit and cooperation. But at the same time, I think, especially in the US and North America in general, where these are still relatively new products and lots of small artisans starting up, I think the more we can collaborate, the more we can share, the better we'll be in the long term.
Absolutely agree. Great points. Okay, we had a question about the interplay between the specific gene sequences versus your developmental or environmental factors with yeast. How much do we know about that? How much can you attribute a certain strain to its sequencing or its environment?
I mean, it's a great question too, because going back to the limitations of genome sequencing, in many ways, it's just the genome tells you the collection of genes an organism has, and those genes will be expressed and they'll behave differently depending on the environment, right? So for cheese, for example, a yeast that's growing at the early stages of making a rind on camembert, that yeast is experiencing a very acidic, a very salty environment. But over time, as a community of microbes develops around it, the pH will go up, it'll become less acidic. Some of that salt will diffuse away. And so we see in my own lab, when we do what's called transcriptome sequencing, so actually looking at the expression of genes, not just the collection of genes, but you see very dramatic changes in gene expression in fermented foods over sometimes even hours. So there really is a dynamic change over time that happens. The problem with transcriptome sequencing, it's a lot more expensive. It's actually harder to get the data. So much of what we have is just genomic sequencing. But once you've landed on a particular strain of interest in a particular product and you want to understand the dynamics of that product and that strain, that's when you could do things like transcriptome sequencing or proteomics or metabolomics, where you can actually look at the proteins or metabolites as well. So yeah, there's a lot of other technology out there we didn't talk about today that can help illuminate that.
Yeah, they are incredibly responsive to their environment, and in addition to genomics and proteomics, I would also say, sensory, smell what's going on, and the yeast is going to tell you. I think that's incredibly valuable as well. Like, you can smell a stressed out yeast, you can smell a stressed ferment because of some of the metabolites that it makes, right? Like maybe it's a little bit sulfury or, eggy, right? And that's important as well. They do signal things that we can detect without sequencers, although like that's extremely useful information as well.
Okay, so building off that question about the environment, so we had another question. If yeast get contaminated by some of those atmospheric microbes, like whatever's in the air, how do you keep these strains, pure? You mentioned that Norwegian yeast. How do you keep it separate from others?
Yeah, I mean, the lab nerd in me says, you check for contamination, right? There's tools that you can use to check for contamination and make sure that that's not there. But if you don't have the lab tools, then it's a little bit trickier. But one thing that we see, and this isn't limited to beer yeast, is that yeast does adapt to its environment, and the yeast that adapt to that environment get really, really good at thriving in that environment. And they often naturally can outcompete some of the, new challengers that might emerge as contaminants. So in some cases, the yeast is pretty good at keeping contaminants out naturally.
Do either of you find that, Richard at Escarpment, Ben at Tufts, do you find wild yeast entering your lab?
Yeah, it's a great question I saw in the chat. We do, and it's really annoying sometimes because, one of the things in a microbiology laboratory you want to do is keep everything really controlled and everything pure, and you want the right things in the right place at the right time. And one thing we find is actually right around this time of year in the last month, trees are leafing out in New England. There's a lot of just life exploding everywhere. And we often find at this time of year, as well as in a lot of fermented food production facilities, you get contamination. And we get contamination by a yeast. It's a group of yeast called the red or pink yeast, Rhodotorula or Rhodosporidium. They're kind of everywhere. They live in water. And if you ever are making, like a cheese, or even at home, sometimes if your cheese turns like pink or red, sometimes it's because of this yeast. And we find it just loves living on leaves. So at this time of year, it explodes. But generally, if we're being really clean in the lab, we don't see a lot of contamination. But it happens. Even in very sterile environments, you still get contamination.
Yeah. Everything's everywhere. There's always, we have methods to prevent cross-contamination, but it always happens. So, from our perspective, you just always have to check, like whether it's a commercial propagation or whether it's an experiment. It's, you have to check and make sure that whatever it is you've grown is not cross-contaminated with something. And yeah, we get some, wild yeast popping up. Like for us, it's always like fast-growing, whatever yeast that can grow really fast on glucose, that'll pop up in growth media every so often. And then we have to, try to make sure that we're able to detect that.
This actually might be a really great time too to note that we have some upcoming webinars related to these kinds of topics about managing microbiomes. I don't know, Amelia, if you were going to give a shout out to these, but on June 16th, Maria Marco and I are going to talk about some of these kinds of broader questions about not just yeast, but all the microbes and microbiomes. So there'll be a webinar called Measuring and Monitoring Fermented Food Microbiomes. And then in July, on July 14th, we're going to talk more about specifically managing microbiomes to deal with these kind of quality issues, like yeast getting in that you don't want, or other kinds of quality issues. So definitely check those out if you're interested in those kinds of questions.
Yes, thank you, Ben. We are taking a deeper dive into these. That leads perfectly into my last question for both of you. So do you see the future of yeast in beer and in other fermented food and beverage? Do you see it lying in, gene editing and strain improvement, or the isolation of wild yeast? What provides more potential?
I think a combination of both, right, Richard? Yeah.
Yeah, why not both?
Yeah. I mean, I think in some ways what we could do is go out, find some wild yeast that maybe have completely new properties, like maybe make some crazy flavor that's never been in, sourdough before, or never been in, an ale or something like that before. And maybe it'd be really hard to grow that specific yeast, making that particular compound in the ferment. Maybe it just won't grow for some reason because it's not adapted to, the sourdough environment. But what you could do potentially is take the gene that is making that flavor, like the example Richard talked about earlier, and actually edit that into some traditional yeast that would normally be found in that fermented food. So that's an example, like he talked about some of the earlier beer examples of getting plant compounds into brewing yeast. So it would be the same kind of approach. But yeah, I think the future will be a mix of those various approaches. Anything else, Richard, in your crystal ball?
No, I would agree. Like you kind of have to have both, right? Like we can't stop looking at what is already out there, either in the wild or in overlooked fermentation cultures, but also, there is this huge potential of taking what we know and creating new applications, whether that's through strain development or whether that's through genetic modification and, other things like that.
Well, Richard, thank you so much for chatting with me today. This has been amazing. I really appreciate the opportunity to think about these really exciting questions with you. And I'll leave it to Amelia to wrap us up.
Yes, thank you. Thank you so much, Richard and Ben. And thank you to all of you for attending today's webinar. We will be posting a recording on TFA's website in the next 24 hours. As Ben mentioned, we have a number of really great webinars coming up in the next few weeks, including measuring and monitoring fermented food microbes. We're bringing Ben back and Dr. Maria Marco and managing microbes to control quality. We also have a webinar coming up on Beyond Kombucha, pu'er, June, and dark tea. So please go to fermentationassociation.org to check these out and register. And while you're there, subscribe to our YouTube channel. Thank you so much.